Am I audible to all?
Yes,
Yes, madam.
Yes, ma'am.
Yes, madam. Good morning.
Yes,
Thank you
madam.
so
Good
much.
morning.
Thank you. So I'm welcoming you all to the today's webinar. I'm Sushri Shwarvatika, a senior research assistant at Heredity Biosciences and today's webinar coordinator. Due to some health issues, our esteemed convener, Dr. Gopal Krishna Purohit, is unavailable today. I will be taking the lead and doing my best to ensure a smooth session. Please bear with us if there is any mistake happen. So everyone knows the today's topic, foodborne diseases, pathogenesis and diagnostics. So I'm delighted to introduce Heredity Biosciences, a premier institute dedicated to training, research, services, placement opportunity in the field of bioscience. At Heredity Biosciences, we proud ourselves to providing hands-on training using instruments like PCR, qPCR, spectrophotometer, microtomes, gel electrophoresis. We have earned the trust over 1,000 students who have shared their positive experience through extensional reviews. Currently, our highly popular summer internship program is going on, and I invite you all to explore the opportunity available with us. Before diving into the session, I am briefly sharing the training course modules and the services we are here at Heredity Biosciences. We offer a variety of specialized hands-on programs designed to equip students with practical skills and industry-relevant knowledge. Our programs include clinical microbiology and genetics, molecular biology, plant biotechnology, environmental technology, food microbiology, nanotechnology, industrial microbiology, and many more. There is some specialized modules also like the real-time PCR, clinical embryology, bioinformatics. And then coming to our service part, in the services, we are providing the Sanger sequencing, the RT-PCR, whole genome sequencing, next generation sequencing, some analytical services like LCMS, GCMS, HPLC, XRD, and FTIR. And then some phytochemical services, microbiological services, enzymatic assay services, proteomic services, that is many more services we are providing here. And specialized highlight is the hands-on training. We are providing complete hands-on training to the students. These are our recent placement activities going on in Heredity Biosciences. More than 200 students joined in different healthcare professionals. So now, coming to our webinar, Foodborne Diseases, Pathogenesis and Diagnostics. Our esteemed speaker, Dr. Priyambada Singh. So it's now my pleasure to introduce today's esteemed speaker, Dr. Priyambada Singh. Dr. Singh is currently serving as an assistant professor in the Department of Microbiology at Kalinga University, Raipur, Chhattisgarh. She holds a PhD in microbiology and has been recognized with a gold medal for her academic excellence. She has successfully qualified CSR UGC NET, GATE, and CGSET examinations showcasing her deep subject mastery. With over 10 years of academic experience, she has made significant contribution through teaching, research, and mentoring MSc and PhD students. Dr. Singh's research interests include medical microbiology, antimicrobial resistance, immunology, virology, and the study of antimicrobial compounds. She has also earned many NPTEL course certification, including being a topper in the course of Introduction to Professional Scientific Communications organized by IIT Kanpur. Dr. Singh has presented her research at various national and international conferences and has published original research articles in reputed journals worldwide. We are honored to have her with us today, share her valuable insights on foodborne diseases and their diagnostics. So here is our team members at Heredity Biosciences. So thank you once again everyone to join us today. We hope you found today's session informative and inspiring. Now, I request our esteemed speaker, Dr. Priyambada Singh, to kindly take over the virtual podium and lead us into the main part of today's session. Ma'am, please.
Hello, ma'am. Am I audible to you?
Yes, ma'am.
Good morning, everyone. I'm Dr. Priyambada Singh, Servicing Assistant Professor in Department of Microbiology in Kalinga University. Today, I'm very glad and thank Heredity Bioacademy for inviting me on this platform and giving me opportunity to give lecture to your students. So, I will be beginning with my lecture. Just wait a minute, I'm presenting my screen. Yes, students, are you able to see the slides?
Yes, ma'am.
Yes, ma'am.
Okay, so today we will begin our lecture about foodborne diseases, their pathogenesis and diagnostic. So the purpose of choosing this topic is that because we are in a season in which this temperature is high and humid, and since we are living in a tropical country, so this tropical environment and the humid condition promotes the growth of various microorganisms, specifically pathogens, who has a optimum temperature of 37 degrees Celsius. So this temperature is very good for the growth of different pathogens and causing different diseases. And since we are about to have these summer vacations in the upcoming months, so people used to travel here and there and goes to different places, eat different kinds of food, and came in contact with different people. So they carry infection via contaminated food, water, or vomits. So that's why I chose this topic to introduce you with the different foodborne diseases, their pathogenesis and diagnostics. And along with this, I will also discuss the different latest techniques which are used to diagnose these foodborne disease before the onset of the infection. So here are the contents which will include the introduction, the common contaminants, foodborne infectious diseases, which will include salmonellosis, sigillosis, diarrhea, campylobacteriosis, botulism, cholera, foodborne viruses, mycotoxin, and detection of foodborne pathogens. Since we are having less time, and this foodborne pathogenesis and diseases is a very vast topic which include various microorganisms, include bacteria, fungi, protozoa, viruses, which are causing different foodborne infections. So I will be including only few and important pathogens which are responsible for causing different foodborne illness. So I will begin with the introduction. Foodborne disease is an illness or a health condition brought upon by consumption of contaminated food. So if we consume a contaminated food, due to that contaminated food, if our health condition is hampered and we face any pathological damage, that is known as a foodborne disease. And similarly, if that disease is occurring in more than two individuals due to same food, that is known as a foodborne disease outbreak. For example, if some person goes to restaurant and they are serving rice, which is 2 or 3 days before cooked, and they are just warming that and giving to different people, and by the next day, most of the people who ate food over that restaurant got infected and have diarrhea, vomiting, and other symptoms. This is known as a foodborne disease outbreak, which is actually occurring in different people due to ingestion of a common food. And this is very recently in Pune, it happened that in the hostel, many students face this foodborne disease outbreak due to consumption of contaminated food. This foodborne illness impact the entire world and especially in United States. Based on recent information from Center for Disease Control and Prevention, annual incidences of food-related disease involve 76 million cases, of which only 40 millions can be attributed to known pathogens. Otherwise, other are still unknown. These foodborne disease results in 3,25,000 hospitalization and at least 5,000 deaths per year around the world. And the frequency and the significance of these foodborne illness depends on interaction between a particular foodborne pathogen, the host in which it is causing the infection, and the environment in which the host and that particular food is present. The majority of reported foodborne illness outbreaks are caused by known pathogens such as norovirus, which is a common virus causing foodborne illness. Then we have Campylobacter, Salmonella, Listeria monocytogenes, and Shiga toxin with producing Escherichia E. coli. To tackle this foodborne disease outbreak, the WHO has recently released a global strategy for food safety 2022 to 2030, which will address all the emerging challenges which we are facing in context to foodborne diseases. For detection of these diseases, including the conventional techniques and the new technologies and innovative approaches to strengthen the food safety system and enhance laboratory capacity for foodborne disease surveillance. So first of all, for preventing any disease outbreak, it is very important to have a proper surveillance of a disease. For example, if a disease is occurring like a cholera or salmonellosis is occurring in a particular area, so there should be proper management of this disease and surveillance by various laboratory means in which pattern that disease is occurring in different parts. Okay, so that if proper surveillance will be there, then proper safety measures could be used, which will be helpful in preventing many foodborne diseases. So these foodborne illness especially occurs when a pathogen is ingested with food and it establishes itself in a human host. Okay, so when pathogen enters the human host, either it will go into your body and then that pathogen will invade your body, multiplies, take the nutrition from your cells and cause the pathological effect. This is the first approach. Second approach is that pathogen will contaminate the food and release its byproduct in the form of toxins into the food, which will contaminate that food, and then we will consume that contaminated food. That's why we classify these foodborne illness into two categories: foodborne infection and foodborne intoxication. Usually the foodborne infection will take long time due to incubation period of organism, but foodborne intoxication will not take time because toxin is already released in the food material which we consume, and according to its release into our body and action, it can act within few hours also. Okay, so all these foodborne disease are associated with poor hygiene practices that we all know. Okay, so whenever we come to home, we should always wash our hands because we have touched different surfaces and we don't know what kind of organism or what kind of spores were at those surfaces. Okay, so whether by water or food transmission, especially the fecal oral route is the key, with the food providing the vital link between the host. Okay, so many people you have seen some fruit vendors, some vegetable vendors, when they are selling their food products like vegetables and fruits, they are washing their food or fruits and vegetables with the water just taken out from the drains. Okay, this news has been released a few years ago that a regular fruit vendor was washing the fruits with the help of the water taken out from the drains. Okay, so these kind of things, because drains have the fecal matter in them, waste material, human waste, municipality waste, every waste is there. Okay, so the When these fecal matter are present in the water sample, and then this water is used to clean the vegetables. So that's how these organisms will reach into the fruits and vegetables. And one more thing, formats such as sink faucets, drinking cups, and cutting boards also play a role in maintenance of fecal oral route of contamination. Fecal oral route means any any food product which is now contaminated with any format which has been contact with the fecal matter anyhow. Okay, either without washing hands, either with the tissue papers, either with the help of water, by any means. If the fecal matter is entering into the food substance and then we ingest it, it will definitely cause any foodborne infection. The food products that has been incriminated in the most outbreaks are poultry, ground meat, seafoods, milk and dairy products, and fruits and vegetables. The typical symptoms of every foodborne illness include abdominal pain, diarrhea, vomiting, nausea, fever, and if leads to complication, it will also lead to respiratory difficulties, and in severe cases, it will also lead to death of a particular patient. So we will divide these foodborne diseases into food intoxications and food infection. So I will explain this chart. You can see, first we will talk about food intoxication. Here you will see various animals like puffer fish. Okay, that is a balloon fish. It contains many poisonous substance. Okay, so that is a poisonous fish. Again, we have amentia. Amentia is a mushroom which is a poisonous mushroom. Okay, due to consumption of that poisonous mushroom, if we face any disease, it could be a foodborne disease, or due to infestation of a chemical like pesticide like chlorpyrifos, DDTs, and heavy metals like arsenic, lead, mercury into the food products, it will also lead to food intoxication. Then we have our microorganisms. These microorganisms which are releasing the toxins primarily includes mycotoxins which are released by fungi. Then we have saxitoxins which are released by algae. Then we have bacterial toxics. These bacterial toxins can be divided into two categories, emetic and neurotoxic. Emetic usually which causes the vomiting like Bacillus cereus. Neurotoxins which will impact the nerve conduction, that is Clostridium botulinum. This could also be a diarrhea like Staphylococcus aureus or Bacillus cereus, or enterotoxins which is again produced by Staphylococcus aureus and Clostridium perfringens. All these toxins are released into the food material, and when this food material is consumed, these toxins will cause the pathological damage into the body. Next, we have food infection. These food infection can again be divided into two categories, toxic infections and infection. Toxic infection means when a toxin producing bacteria is entering into our gut, and along with its growth and multiplication, it is also producing toxin like enterotoxin, then they are known as toxic infection. It includes Vibrio parahemolyticus, Vibrio cholera, Yersinia, Bacillus cereus, E. coli, and different organisms also. Then there is another toxic infection which is a neurotoxin like Clostridium botulinum, which will cause infant botulism. Next are the infections which are due to the actual growth and multiplication of pathogen inside our body. They are taking nutrition from our body, they are infecting our cell, they are taking the nutrition from the cell and damaging the normal functioning of the cell. Okay, due to which there is a less assimilation of food or water by our body, resulting into diarrheal illness, which include intestinal mucosa by Salmonella, Shigella, systemic infection, which includes tuberculosis, Enterobacter species, Listeria, and other infections also like liver, muscles, or fetus, which are infected by different viruses like hepatitis A, Listeria, and some protozoa like Toxoplasma. So this is an overall summary chart which is showing how different foodborne diseases are occurring in different types of hosts based on their intoxication or their infection. So in this chart, we have so many organisms, okay, but I'm going to deal about few and major foodborne diseases, okay, especially the bacterial one. So if we talk about food contaminants, what are actually contaminating our food? Okay, which particles, which matter, which organism are actually contaminating our foods? So these food contaminants are basically divided into three categories. First are the microbial pathogens. Okay, these microbial pathogens will grow into the food like bacteria, virus, different parasite. Okay, and along with this, we will consume the food. They will get into our body and cause infection. Or next contaminant is the toxin. These toxins can be released by animals like mushrooms, algae, fungi, anything. Okay, and then when this toxin enters in a body, then it causes the disease. Or we have different chemical agents like pesticides and heavy metal, which will infect our body. So basically, we divide these food contaminants into three categories: either microbial pathogens, toxins, or chemical agents. Any of one of them can cause any foodborne disease. Okay. So if we see, this is a group of foodborne pathogens. It includes different flatworms like Fasciola, Fasciolopsis, different tapeworms like Taenia, which we are studying from the childhoods, like Taenia solium, which is a tapeworm. Then we have studied roundworms like Ascaris. Protozoa is there, Entamoeba histolytica, which will cause amoebic dysentery. Then we have Toxoplasma, different fungi which are mycotoxin producing like alpha toxins, fumonisins, ochratoxins. Then we have different bacteria. Again, bacteria are divided into gram-positive bacteria, gram-negative bacteria. These gram-positive bacteria which are actually affecting and causing the foodborne infection, which are releasing toxins are Staphylococcus, Bacillus cereus, Bacillus anthracis, Clostridium botulinum. And the gram-negative bacteria, majority of them, which are actually causing the infection, are Salmonella, Shigella, Escherichia, Vibrio, Campylobacter. Viruses include hepatitis A, norovirus. Prions include Creutzfeldt-Jakob disease, new variant forms there. And we have also toxigenic phytoplanktons, which will cause paralytic shellfish poison or domoic acid. So all these are the group of foodborne pathogens. We are going to discuss few of them. So what are the food products which are being infected or being contaminated by different organisms, and what kind of disease is caused by? So if there is food like meat, fish and eggs, or poultry or dairy products, due to these food products, salmonellosis will occur. This salmonellosis will be caused by either Salmonella typhimurium or Salmonella enteritis, which are actually releasing the enterotoxin and causing colitis with the incubation period of 8 to 48 hours. Then we have Campylobacteriosis, which is actually causing the contamination of milk, pork, poultry products. It is the causative organism is Campylobacter jejuni. Its incubation period is 2 to 10 days and it releases a heat labile toxin. The diarrhea, actual colitis, is caused by raw milk or undercooked ground beef. The causative agent is E. coli, which has an incubation period of 24 to 72 hours and called hemorrhagic colitis. Then shigellosis is a disease which is actually caused due to consumption of contaminated egg products, puddings. The causative organism is Shigella sonnei or Shigella flexneri. Its incubation period is 24 to 72 hours. Then due to tofu, milk or meat products, yersiniosis may occur, which is caused by Yersinia enterolotica. Its incubation period is 16 to 48 hours. Vibrio, the cholera, okay, it is actually, many times it could be contaminating seafoods. Okay, the organism is Vibrio parahemolyticus. Incubation period is 16 to 48 hours. Again, this Vibrio will also cause enteritis. Okay, so this is the some foodborne infectious disease, their causative organisms, food products which are affected, and their incubation period. Now we will start different foodborne disease and we will study how these different foodborne infections are actually occurring in our body, their pathogenesis and their diagnostics. Okay. So salmonellosis results from the ingestion of variety of Salmonella cerevisiae, particularly Typhimurium and Enteritis. This group of Enterobacteriaceae have pathogenic characteristics and are one of the most common cause of enteric infection. This bacteria is named after scientist Dr. Daniel Salmon, who isolated the first organism Salmonella choleracis from intestine of pig. The CDC estimates that more than the 1 million people in US contact Salmonella each other with an average of 19,000 hospitalization and 380 deaths. The Salmonella species live in intestine of most livestock and many wild animals. Its infection usually occurs when a person eats food contaminated with the feces of animals or human carrying the bacteria. From the time of ingestion of food, symptoms usually develop in 12 to 14 hours, although the shorter or longer times have been also reported. If we see the pathogens, once we have contaminated that food, what will happen to our body? Okay, so for example, if we ingest Salmonella typhi or Typhimurium, first of all, it will go to our gut. From our gut, some organism will lead to mesenteric lymph nodes, which will cause lymphadenopathy. Then it will be disseminated via blood and reaches to reticuloendothelial system, causing infection in liver, spleen, gallbladder, and finally it reaches to its favorite place, that is pear patches. Okay, where it will grow, multiply, and causes the infection spread to different tissues. Okay, and it will be also expelled out or exit our body via feces. Then it is also present in our blood, causing the bacteremia and generalized infection. During its period from reaching to reticuloendothelial system and excreting out via feces, it is actually showing the symptoms of this enteric fever, including nausea, abdominal pain, and diarrhea. And the period from its ingestion to reaching to the blood is known as its incubation period, which is 5 to 14 days. Okay, we have already heard a famous story of a typhoid Mary. She was a food handler who caused infection in many persons. Okay, she was working in a restaurant and where she handled the food, she was already suffering from typhoid. Okay, have Salmonella. Okay, but due to she is having that bacteria and their formats in her hands or body. Okay, she used same hands, touch the food and serve that food to different people. Via her, that food material got contaminated, bacteria got gone through over the food and reaches to different people. And when those people ingested that contaminated food, she was responsible for causing infection in 78 people and killing 5. This highly infectious carrier posed a great risk of public health. That's why she was known as Typhoid Mary. Okay. So once the typhoid occurs, and the classic symptom of typhoid is enteric fever, which is 104 degrees centigrade. It is a high fever. Okay, it can also be a step ladder type of remittent fever, which can increase and then goes down, then can increase. And it also includes other symptoms like headache, chills, cough, sweating, myalgia, and arthralgia. Some people also develop rashes, which are known as rose spots due to erythematosis. Early intestinal manifestation includes abdominal pain, nausea, vomiting, and anorexia, like this phase. This was early infestation phase. The important signs include hepatosplenomegaly, epistaxis, and relative bradycardia, because so much high fever is there and body is having profuse diarrhea. In that case, what will happen? There is a weight loss, and due to which there is epistaxis and relative bradycardia. If it goes untreated or in some person, it will also lead to complications like gastrointestinal bleeding and intestinal perforation during 3rd to 4th week of illness. It can also lead to neurologic manifestation, which is a rare symptom causing meningitis and cerebellar ataxia. Next is the laboratory diagnosis. How we will diagnose whether a person is suffering from enteric fever and how we will diagnose that it is actually enteric fever or typhoid. So first of all, we will do hematology, in which we will see leukopenia and lymphocytosis. As we all know, when there is increase in neutrophil concentration in our body, it indicates that we are definitely having a bacterial infection. Then we will do the bacterial culture, in which we will collect the sample, either blood, urine, feces, or any patient discharge, and then we will grow it over selective media, that Wilson and Blair media, which will show black colonies, deoxyfolate agar, which will show non-lactose fermented colonies with black center, and McConkey agar. You can see on Wilson and Blair media, it is a selective media for Salmonella, in which the media is yellow in color and Salmonella colonies will be showing black colonies. Okay, in the McConkey agar, these are non-lactose fermenters, and in the XLD agar, these are red colonies which have black margins. Okay, this bacteria can be also cultivated in enriched media like selenite F broth and tetrathionate broth. In the gram stain, we will observe gram-negative rods. You can see this is a gram stain image of Salmonella. In the biochemical test, it shows catalase positive, it is oxidase negative, indole negative, citrate either positive or negative, or urease test. So we have a particular section of biochemical test which we perform for enteric bacteria. Serologically, the most important test which we perform for Salmonella is Vidal test, and we will also do the antimicrobial susceptibility testing. So the antibiotic against which it is sensitive, then we will prescribe particular antibiotic. Okay. Now we have a quick view over the Vidal test. Vidal test is a specific test, serological test, in which we will take patient serum and detect whether antigen O or H from Salmonella is present or not. Okay, so antigen O is the somatic agent antigen of Salmonella. Antigen H is the flagellar antigen of Salmonella. Okay, so we will take patient serum. Okay, and then we will take antigen O and H, when we will dilute it to 1:20, 1:40, 1:80, this dilution in the gradient. Okay, and then we will add the patient serum. Okay, and then we have antigen O and paratype ABC. We will add antigen, we will add antibody, and allow them for agglutination. It is a type of tube agglutination test. Okay, so in the dilution, where we will find a agglutinate, which will indicate a positive test, and further after the dilution, the agglutination will be there. So this is a titer value, means 1:, if suppose agglutination comes here, it shows a positive reaction, means it is an end point and a titer value, that amount of antigens are present in that particular host. So it is a tube agglutination test for detecting Salmonella paratyphi and its antigens. Next disease is sigilosis, which is also known as bacillary dysentery. So dysentery, it is a very dangerous, because from day to two, it will lead a profuse diarrhea, weight loss, and also may lead to death of a person in severe cases. Okay, this bacillary dysentery is quite similar, have symptoms along with the amoebic dysentery. Okay, but it has some difference. So shigellosis or bacterial dysentery is a diarrheal illness resulting from an acute inflammatory reaction of intestinal tract caused by Shigella. The Shigella bacteria has 4 different species: Shigella flexneri, Shigella boydii, Shigella sonai, Shigella dysentery. This Shigella is restricted to human host. And how this organism is transmitted? It is transmitted by 4F. 4F means either food, finger, feces, or flies. Okay, so these 4F are the major cause of transmission of bacillary dysentery. Okay, finger, food, feces, and flies. And it is more prevalent among the children, especially 1 to 4 years of age. It is also having antigen, somatic O antigen, toxin, endotoxin, enterotoxin. It has an important Shiga toxin, which is a neurotoxin present in bacterial cell. So if we see the pathogenesis, Shigella are intracellular parasite, means they will grow inside your cells that multiply within the villus cell of colonic epithelium. The bacteria induce the pair patches cell to phagocytosize them. After being ingested, the bacteria disrupt the phagosome membrane and are released into the cytoplasm where they reproduce. Then they invade the adjacent cell. So you will see here, once Shigella is ingested via contaminated food. Okay. I will take a small pause. Students, are you able to see this picture?
Yes, ma'am.
Okay. So, when we ingest Shigella along with the contaminated food, it will go into your gut where it will be having resistant to bile salts. It is also resistant to acidic pH of stomach. Then it will invade the colonic epithelium. Okay. It will grow there or it will be released via feces. Okay. If we see the section here, this is your colonic epithelium where different cells, okay, these Shigella induce your cell to internalize them, okay, and once your cell internalize them, they will grow inside the cell. For example, macrophage, okay, then this will rupture these macrophage, okay, and then infect the other adjacent cell and it will spread from cell to cell, leads to destruction of, like you can see here, neutrophil destruction of epithelial layer, okay, invasion into epithelial cells. So it is actually inducing your cell to come and phagocytose me, okay, and once your cell phagocytosis is, it will get into the cell and start destructing the cell and start multiplying within your cells, okay, and once infecting one cell, it will multiply and go to the adjacent cell and then infect other cells. As a result of it, Shigella initiate inflammatory reaction in mucosa. Both endotoxin and exotoxin may participate in disease progression, but the bacteria do not usually spread beyond the colonic epithelium. It remains there only. The watery stool often contain blood, mucus, and pus, and in severe cases, the colon can become ulcerated, okay, because they are actually damaging the epithelial cell. So in that case, ulcer will occur naturally. The virulent Shigella produce a heat labile AB endotoxin, which is known as Shida toxin, which leads to kidney failure also. The clinical manifestation incubation period is 1 to 4 days, and during the infection, it typically causes the dysentery. In the initial phase, it will show watery diarrhea with fever, malaise, anorexia, and vomiting. So in the phase of dysentery, there is bloody mucopurulent stool with abdominal cramps and tenesmus. Phase of complication includes in children less than 5 years of age, which will lead to toxic megacolon, perforation, rectal prolapse, and Ekiri syndrome or toxic encephalopathy. So you can see, due to dysentery, how it is progressing and causing severe complications. How it will be diagnosed? For specimen, we will take the fresh stool, and fresh stool we will then prepare a wet mount. In the wet mount, you can see here, Shigella will be observed rod-shaped bacteria. Okay, so it can also be carried via transport media. Sacs buffered glycerol saline broth is used. Wet mount preparation, which will along with the cell, which will show pus cell and erythrocyte. In culture, we will use enrichment broth like selenite F broth or tetrathionate broth. Selective media is same for this salmonella, but the colonies will be of different colors. Deoxycholate agar, xylose lysine deoxycholate agar, Mekongki agar. Okay, but you can see the color of colonies are quite different as compared to salmonella. Okay. In the culture smear motility test, it will show gram-negative bacilli and are non-motile. You can see here, these are gram-negative rod-shaped bacteria. In the biochemical reaction, it will show catalase positive, oxidase negative, indole negative, citrate negative, and urease test. Again, I have also told you that it will be, there is a separate biochemical identification of gram-negative bacteria. Again, we can also do slide agglutination test for Shigella, and also we will perform antimicrobial susceptibility testing. Next bacteria and the next disease is very common, which is known as traveler's diarrhea. It is usually causing in the people which are traveling to many places, and since that E. coli is already present in that environment and resident population is already having that E. coli with them, but they have immunity against that E. coli, so resident population will not have any symptoms, but the people who are traveling come to that area, and then when they came in contact with E. coli, they got the infection. That's why it is known as traveler's diarrhea. This E. coli is a gram-negative non-spore forming rod. It may or may not be motile, and some rods are flagellated and some are not. The pathogenic variants of E. coli, pathovars or pathotypes, all cause much morbidity or mortality worldwide. Many of these pathotypes are major health concern as they have no infectious doses and are transmitted through ubiquitous mediums, including food and water. This transmission occurs when food or water that is contaminated with feces of infected human or animal is consumed. So as I told you, if the resident people are getting this E. coli, they will not get any symptoms because they're already immune to it. But person who is coming from other place, for example, if you went to some place for vacations, those people, if they came in contact with E. coli, they have immunity. But if you go to them and use that food, water, you will get the diarrhea because you don't have immunity against that strain of E. coli. So if you see the pathogenesis, pathogenic E. coli have been categorized into 6 groups according to the pathogenic mechanism. It is enteropathogenic E. coli, EPEC, enterohemorrhagenic E. coli, EHEC, which is also known as Shiga toxin. Then we have enterotoxigenic E. coli, ETEC. We have enteroaggregative E. coli, EAGGEC. Enteroinvasive E. coli, EIEC. Attaching and effacing E. coli, AEC. Why we are naming these pathogenic types in different names? Because their their type of infection in the cells, epithelial cells, is different. How they're actually entering into the cell is different. So I will show you one example. So here you will see the pathogenesis. Here it is different invasions of different pathogen, all 6 forms of E. coli you can see here: ETEC, EHEC, EAEC, DAEC, EPEC, and EIEC. So you can see this is your bacteria and this is your epithelial cell. How they will enter directly, or they show attachment, or they have aggregative adherence in form the mucous biofilm. So their route of entry or their infection procedure or their infestation into the body, into the epithelial cell is different. That's why they are pathogenic forms of E. coli. So I will give you an example like enterotoxigenic E. coli strains produce one or both type of 2 distinct enterotoxins which are responsible for diarrhea and are distinguished by their heat stability, like ST, you can see here ST, okay, heat stable enterotoxin, and LT, which is heat labile toxin. Okay, this ST binds to a glycoprotein receptor that is coupled to guanylate cyclase on the surface of intestine epithelial cells. Upon activation, guanylate cyclase stimulates the production of cyclic guanosine monophosphate CGMP, which leads to secretion of electrolytes and water into the lumen of small intestine, manifesting a watery diarrhea. As soon as the LT binds to specific gangliocytes on the epithelial cells and activates membrane-bound adenylate cyclase, which leads to increased production of cyclic AMP, to the same mechanism employed by cholera toxin. Again, the result is hypersecretion of electrolytes and water into the lumen. Okay, so as a result, both these strains, what are causing? They are causing secretion of electrolytes and water into the lumen of intestine. That's why the person who is having that E. coli, it will be having watery diarrhea. Okay. Next, clinical manifestation. As E. coli is infecting the body, it will cause urinary tract infection. Okay. It may also cause neonatal meningitis when it reaches from mother to fetus. Okay. Or it will be also causing gastrointestinal, which is a prominent disease, diarrhea. Other infection caused by it are abdominal infection, pneumonia, meningitis, osteomyelitis, or bacteremia. For the laboratory diagnosis, again, we will do hematological investigation. In case of bacterial infection, there is increase in concentration of PMNs. For bacteriological investigation, we will take the specimen like urine and feces, and we will culture in eosin blue, which is a selective media for E. coli. You can see it is showing the metallic sheen growth of E. coli in EMB. So it is a selective media for E. coli in which E. coli will show selective metallic sheen, which proves that it is a E. coli. If we culture in McConkey, they are lactose fermented and will grow pink colonies. And in the gram strain, you will show short rods which are gram negative. Okay. We can also do culture smear and motility testing. They are motile gram negative bacteria. Then here are different biochemical identification which are similar, for almost similar, or one or two tests are different enteric pathogens. It is also fermenting most of the sugars and will show antimicrobial susceptibility testing according to the type of strain which is infecting. You will see here, this is indole positive test showing the MR positive test, VP negative and citrate negative. So these are the characteristic features of E. coli. Okay. Next disease is Campylobacteriosis or Campylobacter gastroenteritis. Campylobacters are members of family of Campylobacteriaceae and Campylobacter jejuni. It is one of the common cause of diarrheal illness. It is associated with variety of condition in human ranging from septicemia and enteritis. Okay. Campylobacter jejuni is a slender gram-negative motile curved rod found in intestinal tract of animals. It causes an estimate 2 million human cases of Campylobacter gastroenteritis, which is the inflammation of intestine or Campylobacteriosis. How they are transmitted? Again, through contaminated food and water. Most cases of Campylobacteriosis are associated with eating raw or undercooked poultry meat. Okay, so people who think we can eat raw meat, okay, so you will get Campylobacter in this case, because bacteria is there, because it is already living in the animals. Okay, from the animals, if you don't cook it or it is present in the soil where the animals are feeding or here and there. Okay, unpasteurized milk, contaminated water, and from cross-contamination of other food by these items. There are very different forms of other pathogens associated with foodborne illness that are essentially microaerophilic, growing in West atmosphere, containing approximately 10% CO2 and approximately 5% O2. So they are microaerophilic nature. They require very less quantity of oxygen for their survival. So here there is the pathogenesis. It is extensively found throughout the nature. It can colonize the intestine of both mammals, and transmission to human occurs via contaminated food products. Its virulence factor is, first of all, its motility. You can see Amphitrachus flagellates both the ends, secreting enterotoxin, cytotoxin, which is known as CDT, cytolethal distending toxin. Okay, it is actually a cytolethal which will cause lysis of the cells. Okay, it invades the epithelial layer by first attaching to epithelial cells, then penetrating through them. Diarrhea results from damage of epithelial cells. Systemic infection can also occur, causing more severe illness. So you will see here, it is reaching the target size, it will adhere, it will invade the cell, releases the toxin, CDT toxin, cell damage via toxin and protease production. Okay, you can see. And then it will show antimicrobial resistance genes. It will also have a biofilm factor and efflux pump to prevent itself from antimicrobial compound. So you can say it is disrupting the epithelial barrier, and once epithelial barrier is damaged, okay, the inflammation in the colon will start. Its clinical manifestation includes intestinal infection, inflammatory diarrhea, abdominal pain and fever, extraintestinal due to Campylobacter fetus in immunocompromised individuals only, okay, which will lead to bacteremia, sepsis, meningitis and vascular infection. Especially the person who have a HLA of B27 phenotype, they can also have a reactive arthritis due to Campylobacteriosis. Okay, it triggers the pathogenesis of various other diseases such as Guillain-Barré syndrome and alpha chain disease. Okay, so it is responsible for triggering pathogens of other diseases also. For the laboratory diagnosis, we will do direct microscopy, gram staining. You can see in the gram there are curved rods, gram-negative bacilli, S-shape or spiral shape. Okay. Dark ground microscopy, darting motile bacteria will be seen. You can see this is a scanning electron microscope image of Campylobacter. It is a slightly curved bacteria or a spiral form having the polar flagella. The transport medium for this bacteria is Carey-Blair medium. The selective media, we have faces or rectal swabs which are plated onto the sucrose selective media or Campi-BAB selective media. The culture condition is a microaphilic condition which includes 5% oxygen, 10% because it is a capnophilic organism. It requires carbon dioxide but less oxygen for its growth. Also, they grows at 42 degrees Celsius. Thermophilic Campylobacter, Campylobacter jejuni and Campylobacter coli. In the biochemical identification, along with the other tests, it will show positive nitrate reduction test, hippurate hydrolysis, and it will grow at 42 degrees Celsius. So these are the selective tests for biochemical identification of Campylobacter. Next, an important disease is botulism. It is very important disease because it is used in biological warfare as a bioweapon. Okay. Botulism is a disease in which Clostridium botulinum releases a toxin, which is a botulin toxin. It is a neuroparalytic disease. It is caused by potent toxin released from Clostridium botulinum. Three major types of botulism is there: infant, foodborne, and wound. It is a potential bioweapon due to its potency. The bacteria is gram-positive rod. They are spore-forming anaerobic bacteria, produces toxin that can cause botulism. 7 neurotoxic subtypes are there, which are labeled from A to G. This bacteria is first recognized and isolated in 1896 by von Edminger. So this bacteria is actually producing, but bacteria is not causing anything, but it is releasing a toxin, which is a botulin toxin, which is actually a neurotoxin. So how it is transmitted? A variety of food products such as canned corn, peppers, greens, beans, soups, beets, asparagus, mushroom, ripe olives, spinach, tuna fish, chicken, chicken livers, liver pate, luncheon meat, ham, sausage, stuffed eggplant, lobster, and smoked and salted has been associated with botulinum toxins. Especially, this disease arrived from the production of toxin by Clostridium in canned food. Okay, when due to improper food preservation or food safety standard, following the food safety standards, sometimes Clostridium grows over there and releases botulin toxin in the food substance. These Clostridium botulinum are motile by means of peritrichous flagella and produce botulinum neurotoxin, the most lethal poison known till now. There are 7 types of botulinum neurotoxin A through G based on antigenic specificity of toxin produced by each strain. Okay, once released into bloodstream, the botulin toxin irreversibly bind to acetylcholine receptor in the neurotransmitter junction. Okay, directly it will go to your neuromuscular junction and bind irreversibly to the acetylcholine receptor. It alters the mechanism of acetylcholine release, making the neuron unresponsive to action potential. So, by seeing this, we can imagine when it is actually stopping the nerve conduction, and when it is stopping the nerve conduction, it will lead to progressive paralysis. So, you can see it is a normal neurotransmitter release. You can see this image. Okay, this is one synaptic cleft. Okay. These are the vesicles which are having neurotransmitters, okay? And these are the muscle fibers. Once neurotransmitter are released, then they will go to acetylcholine receptors, and this acetylcholine binds to it and carry out the nerve conduction to other nerve. This is how signal is transferred from one neuron to another, and our body show movements, okay? But if there is botulin toxin is there, what will happen? This is your botulin toxin. Okay, It will go there. You can see snare proteins. And now this vesicle will not be able to fuse here. That's why no neurotransmitter released. And how much receptor may be present here? No neurotransmitter release, so there is no reception. If there is no reception, there is no nerve conduction. That's why it is stopping the nerve conduction. As a result, this muscle fiber is paralyzed. So, what are its clinical manifestation? The most important, due to contaminated honey, okay, and ingesting contaminated foodstuff, lack of breast milk or untreated natural honey or household dust which contain Clostridium spores, infant will got this botulism, okay, which afflicts babies from 1 week to 1 year. Antigenic variation is type A to B. As a result of, because we know as soon as it get into the body, it will go to the nerve conduction and stop the nerve conduction. And when it is actually stopping the nerve conduction, what will happen? What child will be having? Overfeeding, because muscles are not moving. Weak gag, weak cry, decreased movement, appear lethargic, it will flat, blunt, it will not show any expressions, trouble swallowing, excessive drooling, muscle weakness. So you can see all things which are abnormal for a normal baby, which will occur. It will not show any movement. You can see the picture here. Okay. But for this treatment, recovery new drug baby big, which is a botulinum immune globulin, which is a passive type of immunization given to baby. Okay. So it will cure this disease and relieve the symptoms. Okay. It drastically reduces lethargy, IV feeding, and overall hospital stay with early detection, proper treatment, and no long-term effects will be observed. Okay, so as soon as the baby is showing these symptoms, if we take them to healthcare practitioner, they will recognize the infection. Okay, and they will give the botulinum immune globulin, so we can save the baby. Okay, and we can reduce all the symptoms which are in the baby. But if we delay, then it will cause progressive paralysis, may lead to death of that baby. Other is foodborne botulism. It will happen in adults also, caused by eating food containing toxin produced by Clostridium botulinum. Okay, I will tell you a story. In old time, not so much old time. So during the wars, what people used to do? Okay, they spread the botulinum toxin into the water bodies. Okay, so the soldiers from other country when come there and they consume that wood, they get paralyzed and get killed. Okay, so it was used as a bioweapon from the long time. Okay, in the old time also. It is non-commercially canned food at least higher risk for being contaminated. Sign and symptoms is 12 to 36 hours after muscle weakness, diarrhea, blurred vision, vomiting, and eventual paralysis. So if a person consumes that toxin containing food or water, okay, progressive within 12 to 36 hours, muscle weakness will be there, diarrhea will be there, blurred vision, vomiting, eventual paralysis, and then whole body is paralyzed and lead to death of that person. Okay, so it was already used in wars and also used as a bioweapon. For the laboratory diagnosis, gram-strain smear of suspected food products, which will have gram-positive non-capsulated bacilli with subterminal spores. You can see this is the subterminal spores, and it is a gram staining of Clostridium botulinum. If they are motile peritrichate flagella, in the blood agar or Robertson cooked meat broth, it will grow. You can see these type of colonies over the blood agar, and this is a Robertson cooked meat broth. It will convert that meat into black color with a foul odor. Okay. For the toxin demonstration, we will use mouse bioassay. We will inject toxin into mouse and we will see the progression of paralysis. This is how the botulinum is being diagnosed. Next disease is cholera. It is very common disease caused by Vibrio cholera. Once the bacteria enter the body, they adhere to intestinal mucosa and they are not invasive but secrete cholera toxin, which will cause prolific diarrhea. It is a gram-negative comma-shaped bacterium. It is a facultative anaerobe and have a flagellate one end of the soul. When one cell pole as well as pili and undergo respiratory and fermentative metabolism. When this cholera toxin or Vibrio cholera is ingested, it can cause diarrhea, vomiting in a host within several hours to 2 to 3 days of ingestion. It has again having two antigen, O somatic antigen and H flagellar antigen. It is having a collagen, enterotoxin, and endotoxin. This enterotoxin is having two subunits, A and B. So here you can see the pathogenesis when it is entering, transmitted via contaminated food, water, and undercooked food. We can see this Vibrio cholera, two components A and B. It is entering into the cell. Okay, when it's entering into the cell, it will go to the adenyl cyclase, CAMP will be released, and chloride ion will be removed. Okay, so you can see ions are being released, water is also released. So it is resulting into profuse diarrhea. The incubation period is 1 to 4 days from person who develops symptoms, depending on the size of inoculum, how much amount of Vibrio has been or cholera antigen has been ingested. It manifests with nausea, vomiting, profuse diarrhea, and abdominal cramps. The characteristic feature of cholera is rice water stool. Okay, light white color stool is there. Loss of fluid leads to profound dehydration and circulatory collapse may occur in this condition. Okay, so much release of ions or salts or water from the body. Okay, so leading to rice water stool, which is a characteristic feature of cholera. It is actually used in diagnosis of this Vibrio. Okay. So you can see for the laboratory diagnosis, stool sample is taken, grown in VR media and rectal swab point, trypticase, taurocolate, telluride broth, smear examination, you will see gram-negative comma-shaped bacteria. So it is very easy to diagnose this because none other than Vibrio cholera, it is a comma-shaped bacteria. Okay. Then there are motile bacteria, we will do hanging drop method. In the culture, we will see nutrient agar, moist translucent colonies, McConkey colorless colonies, and in the blood agar, green zone around the colonies. You can see this is the green zone around the colonies over the blood agar. Okay, so this is the laboratory diagnosis of Vibrio cholera. We can also use enrichment media like bile salt agar, Monsters green taurocolate, trypticase, telluride agar, biochemical test, it will ferment glucose, sucrose, maltose, mannitol. It is oxidase positive and give a nitrate reduction test. For serological investigation of O or H antigen, we can use indirect hemagglutination, complement dependent vibricidal antibody test. For treatment, we will give oral tetracycline or vaccine. There is a killed cholera vaccine is already given to during the immunoprophylaxis during the childhood. So this is the summary of all bacterial diarrhea and food poisoning. Okay, starting from Staphylococcus aureus, Bacillus cereus, Clostridium perfringens, Clostridium botulinum, E. coli, and hemorrhagic E. coli, enterotoxigenic E. coli, Vibrio parahemolyticus, Vibrio cholera, Shigella, Salmonella, Salmonella enterica, Clostridium difficile, Campylobacter jejuni. Okay, so all this is a summary chart how their incubation period vary from each other. In which disease there is profuse vomiting. For example, Staphylococcus aureus, there is severe vomiting. Okay, in diarrhea you can see Clostridium perfringens food poisoning will cause profuse diarrhea. Clostridium botulinum diarrhea is very rare because it is actually neurotoxin. Okay. In case of Vibrio cholera also, profuse diarrhea, rice water stool is there. Okay. Clostridium difficile, Campylobacter jejuni, they are also causing profuse diarrhea. In all the cases, okay. In case of Salmonella and Salmonella typhi, I have already told you, characteristic feature of typhoid is 104-degree fever, okay, which will like a step ladder fever. Okay, it will goes up, then goes down, then goes up, then goes down. So if person is having high fever with the diarrhea or vomiting, definitely it is a typhoid. We can do direct Vidal test after this. Okay. And you can say for epidemiology, I've already told you that how from different food materials it is entering to our system, and actually they're infecting the instant all organisms. all organisms in one or another way, they are infecting the intestinal epithelium, which as a result, intestinal epithelium are unable to absorb the food or assimilation. As a result, they are releasing the ions and water into the lumen. As a result, they have diarrhea. Okay. Due to extensive growth, person will also have abdominal cramps or vomiting. Next, we will see foodborne viruses. The most common foodborne virus pathogens are hepatitis A and norovirus. These viruses are frequently transmitted via food bivalve mollusks such as clams, cockles, mussels, oysters, and especially prone to transmit viruses. The water in which they grow are increasingly subject to human fecal contamination, sometimes from sewage discharge and sometimes from infected shellfish harvesters. Okay, so the water which is released from the sewage, anyhow it came in contact with the food chain. Okay, the disease or the excreta, anyhow it comes to in contact with the food chain, it will get inside the body and causes the infection. So we will discuss only two viruses here, hepatitis A and norovirus. We know hepatitis A virus particle are environmentally hardy organism that can be transmitted by contaminated food, water, environmental surfaces like contaminated tabletops, cooking utensils, and through direct or indirect person to person. You can see this is a virus, single positive strand RNA genome. Okay, no lipid containing envelope is there. Okay, it will cause infectious hepatitis or epidemic jaundice. It is an acute infectious disease caused by hepatitis A virus, which is also known as HAV. The disease is heralded by non-specific symptoms. So earlier, when it came to your body, there are non-specific symptoms for any foodborne disease like fever, chills, headache, fatigue, generalized weakness, ache, pain all over the body. But as soon as the disease progresses, there will be anorexia, nausea, vomiting, urea, and ultimately leads to jaundice. Why? Because these hepatitis A virus are infecting the hepatocytes. Once you enter in your body, it will go to intestine via there. They are disseminated to different parts, and in the different parts of body, their site of all infection is hepatocytes. Okay. This disease is benign in nature with a complete recovery in several weeks. Then we have norovirus. You can see norovirus are cause the majority of acute viral gastroenteritis cases worldwide. In addition, according to WHO, norovirus is nowadays leading to cause acute gastroenteritis among children less than 5 years of age who seek medical care. This is a non-enveloped virus, you can see, with a diameter of 30 to 35 nanometers, single-stranded RNA genome, and it's spread by a fecal oral spread in primary mode of transmission. Okay, this virus has the ability to withstand a wide range of temperature from freezing to 60 degrees Celsius. So you can see nothing can destroy this virus. Okay, and to persist on environmental surface and food items contribute to rapid dissemination, particularly via secondary spread via food handlers or family members. So you can see this is the structure of this virus. Next are the mycotoxins. Okay, mycotoxins are released by fungus, and the major mycotoxin which is involved in the foodborne infection is alpha toxin. It is a fungal toxin produced as a secondary metabolite by fungi, Aspergillus flavus and Aspergillus parasiticus. These two fungi grow as a saprophytes. They have worldwide distribution but are more common in tropical climates. I've already told you, we are in tropical climate. We are having these organisms around us so commonly. Okay, they have wide variation in rainfall, humidity, and temperature. The commonly contaminate from this mycotoxin is corn, grain, peanut, and other food crops during their growth, harvest, storing, or processing. The exposure to alpha toxin B1 is known to cause both chronic and acute hepatocellular injury and typically leads to liver cancer. Indeed, alpha toxin are extremely carcinogenic, mutagenic, and immunosuppressive as well. So if we see the structure of alpha toxin, you can see We divide this alpha toxin into B1, B2, M1, M2, G1, G2. They have different structures. They are dipyranopomerocyclopentanone alpha toxin. This is the structures. Okay, these are the secondary metabolites and they are extremely poisonous. Okay, they have a fluorescence at 365 nm of UV light and will show green. This one G1, G2 will show green fluorescence. Okay, among these B1, this B1 is the most common and most potent carcinogens. The alpha toxin B1, B2 after ingestion by lactating animals are modified in animal body to yield alpha toxin M1 and M2. So if this B1 and B2 are ingested by lactating animal, it will convert to M1 and M2. If cattle consume alpha toxic contaminated food, alpha toxin also cause appear in milk and dairy product. Okay, so you can see if a cattle is consuming the contaminated food with alpha toxin, okay, and if we take that milk or dairy product from that cattle, it may enter in our food system also. Besides their importance in grain, they have also been found in beer, cocoa raisins, and soya. So how these mycotoxins are detected? So first of all, for the detection of mycotoxins, we will take the food materials only. Okay, we will do analytical method, mycotoxin sensors, and rapid methods. In analytical methods, we will perform TLC, HPLC, and GC. For the sensor, we are using immunosensors for developing antibodies against them. These could be peptide-based, APTA sensor, or MIP-based. Rapid methods include ELISA, LFA, and FPIA. Or either it could be enzyme-based or fluorescent-based. Next, how we will detect all these foodborne pathogens? Okay, because we know foodborne pathogens are causing infection. After causing the infection, we are taking the patient, and from patient we are doing the laboratory diagnosis. Then we will see how patient is developing. But until then, it has already affected the person, has made several loss. So we should make the measures so that these foodborne pathogens could be detected earlier, whether the food is contaminated with particular pathogen or not. So the major goal is maintaining food safety is the rapid detection of pathogenic microorganisms in order to curb outbreaks that can affect large populations. Standard culture techniques may require days to weeks. Obviously, because the culture during the incubation period for bacteria is 24 to 48 hours. Okay, usually it will go for 24 hours, but for doing other tests, we will need 48 hours. So till that 48 hours, it could spread to other people also. Okay. This identification is often complicated by low number of pathogens compared with background microflora. Furthermore, the varied chemical and physical composition of food can make isolation very difficult. Okay, because we are isolating them from contaminated food. So the composition of food will also cause its difficulty for its isolation. Nowadays also fluorescent antibody, enzyme-linked immunosorbent assays and radioimmunoassays have a proven value for detection of these foodborne pathogens. We will take the serum sample from the patient and detect the antigen by them. Okay, these can be used to detect small amount of pathogen-specific antigens. So if we are detecting these foodborne pathogens, so you can see these are the food particles. Okay, we are taking the pathogenic organisms, detection process and detection. But this process, how it can be done in quick manner or in short duration time, so that we can control the outbreak of particular infection. So we have here conventional technique like culture plating, which I've already discussed. Then we have antibody-based immunoassay, PCR-based detection, or MALDI-TOF, in which we will detect the particular either DNA of that pathogen or particular toxin secreted by that pathogen. We are also having emergent techniques like molecular imprinting, DNA microarray, aptamer-based immunoassay, omics or CRISPR-based technologies, integrated biosensing approaches, so that we can have an early detection of these foodborne pathogens. We can either take the DNA and do the microarray using different probes. We can detect these diseases in short duration, or we can do a early detection to prevent several outbreaks of particular disease. So we can also use molecular techniques which are make easy to detect the presence of specific pathogen to detect virus that cannot be grown conveniently, because if there is bacteria, we have a conventional culture technique, but if virus is infecting, then what we will do? Okay, so for this we will use DNA or RNA probes or PCR. So for the detection methods, we have traditional methods, which I've already told you, but the novel methods, nucleic acid based assay, ELISA, latex agglutination antibodies, like we were using immunological immunosensor, enzyme based sensors, biosensor, electrochemical sensor, magnoelastic sensor, optical. So in these, just what we are actually doing, we are taking the antigenic component or a particular component from that pathogen or by a toxin produced by these organisms, and then we will detect the presence of that particular toxin or organism in the food materials. Okay, this is how we are detecting. For example, if there is a canned food, so many canned food, okay, and if particular person is infected by consumption of that canned food, and after 2-3 days that person went to lab, so by seeing the symptoms, the laboratory can make the surveillance what you have consumed, and then they will take those canned food and do the early detection, so that supply of that canned food can be stopped and we can prevent it from breaking out. Okay, having an outbreak. Now we have also next generation sequencing strategies for detection of foodborne pathogens, in which we can do meta-barcoding, full genome sequencing, or metagenomics, by which from the contaminated food we will take the nucleic acid. Okay, we will extract the nucleic acid. Either we will do meta-barcoding, in which we will generate the primer for the target typing region. So after that we will do PCR products. Okay, then mapping, then clustering, and then matching with the known or unknown. Okay, and then we will identify the genotype and we'll say whether it is a previously known organism or a new organism. Either we can also do from this DNA, we can also do full genome sequencing by synthesizing degenerate primers and spanning the genome, making the amplicons, and from these amplicons we do the de novo reference based assembly, and then we will have a partial or full genomes, and then we will identify from which genotype or it belongs, then we will do the strain identification. Or we can do metagenomics. We will take several samples, do the sampling process like filtration, nucleases, selection, enrichment. Okay, or we will directly take those bacteria, those DNA, or we will do de novo reference based assembly and other approaches by which we will generate the DNA. Okay, and after that we will have a partial or full genome sequence, and from then we will do genotypes from the previously known or known primers, from the sequences which are present in different organisms, and then we will able to see whether it is a previously known, or if it is a previously known, we know this contaminated food may be having more than one pathogen at a time. Okay, so this is how by using next generation strategies we can detect any foodborne pathogen in any huge range of contaminated food, or the food which is suspected to be contaminated. This is how we can prevent many foodborne infections. So, major advance in the detection of foodborne pathogenesis, the use of standard pathogen DNA patterns or foodborne pathogen fingerprinting is done. The Center for Disease Control and Prevention has established a program which is known as PulseNet, in which pulse field gel electrophoresis is used under careful control duplicated condition to determine the distinctive DNA pattern of each bacterial pathogen. With this uniform procedure, it is possible to link pathogen associated with disease outbreak in different parts of the world to specific food source. Data from around the world that is used in FoodNet. Okay, you can see an active surveillance network to follow 9 major foodborne disease. Using FoodNet approach, it is possible to trace the course and cause of infection in days, not in weeks. Okay, so there is a network of FoodNet. Okay, it is a surveillance network in which we are using pulse field gel electrophoresis. Pulse field means by the movement of protein or DNA, we will detect the pattern of the DNA, and every organisms, the DNA is having different pattern of movement. Okay, and then we will match it with the known one. So it is already known for the 8 organisms, okay, by which if some food product is generated and distributed all over the world, okay, or distributed in various part of the country at a time, if certain disease is detected in one place via foodnet, it information will be sent to different places via surveillance, so that we can detect that those places, at those places that canned food has been supplied, they will also do the same pulse field electrophoresis and then report to foodnet. This is how early detection of pathogen will be done in days, and we will prevent the disease outbreak. So these are the references from which, like CD Center for Disease Control and Prevention, I have taken many data for this presentation. And these are my latest publication, like next generation sequence disease detection and research and treatment. And I'm also having a book coming for Taylor and Francis group, like harnessing micro for sustainable food system. It is under printing. It will be published soon. So I will be winding this lecture. Thank you. Yes, student, the topic is open for discussion. Any doubt regarding foodborne diseases?
Excuse me, Anna.
Good morning,
One
ma'am.
question. Hello.
Yes.
Hello.
Yes, you're audible.
Most of the microbes are entering into body in contaminated food. Okay, ma'am. Hello. 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 Hello,
Yes,
ma'am.
you're audible. Please say.
Most of the microbes are entering into our body in contaminated food. Our food
Yes.
is digested in our alimentary canal. But why
Yes.
do such microbes, which are pathogen to our body, are not digested?
Because the food are made up of complex biomolecules, and these microorganisms are also made of these biomolecules, but in such manner they have virulence factors. Okay, there is a virulence factors is there. These virulence factor prevent these bacteria from various enzymes which are present in our gut. As we know, when food enters in our body, our gut enzyme cleaves that food, digests that food. Okay, but
Yeah.
when bacteria is there, it has having several like proteases or hyaluronidase, DNases. It will cleave the enzyme. Okay, they will
Okay. Okay.
cleave the enzyme and
Okay.
make their so digestion impossible. Okay.
Okay. Thank you, ma'am. Thank you.
Ma'am, it was truly a great session. You have explained everything in detail.
Thank you so much. The students, any other thing we want to discuss?
I want to ask a question.
Yes, Kamlesh, you're saying something?
What is the major difference between the diarrhea, dysentery, and
Can you
constipation?
please increase the voice? Sound is very low.
What is the major difference between the diarrhea, dysentery, and constipation? How can we detect on that?
Your voice is very low. I just heard the word diarrhea.
Yeah. I want to ask one question.
Can you write it down? Your voice is very low, or you can just
Okay.
increase the sound of your speaker.
Okay, ma'am.
Anyone has any question?
Dr. Deepti here. I wanted to ask if I want to send any food for such analysis, where do you recommend? I mean, as per your knowledge, like you said, metagenomics can be done, or these kind of, if somebody is suffering or they suspect that they are, especially we can detect those viruses and parasites, but those enteropathogenic, all those, how can we send that?
What you want to send, either
Suppose
patient
I want to
sample
send,
or food sample. That is a difference.
Yeah, both can be.
Whether
Yeah.
we can send food sample for detection via metagenomics, or we can first we will detect the patient which antibodies it are having against particular disease. Okay, because if we are doing metagenomics, we need huge quantity of sample, and that sample, from that sample DNA will be extracted directly. DNA will be extracted, and then how many types of DNA is there, they will be matched with the standard ladders. Okay, by having these trimmed primers specifically designed all over the world, so which type of pathogens we are expecting. Okay, for example, if there is diarrheal illness and doctors are thinking it could be diarrhea due to bacteria. Okay, most of the samples are similar to bacteria, so they will not use the primer which were used for viral infections. They will only use the primers which for bacterial DNA. But in case still there is no chance of any bacteria, then they will go to virus.
Okay. So, ma'am, do you recommend that generally we send stool sample or we send vomit sample? Such
Usually
samples?
in our access, because NGS is now in running stage. Okay, so metagenomics is preferably done nowadays.
Okay. Okay. I got your point. Thank you.
Yes.
So if anyone have any questions now, I will share ma'am's mail ID. Through mail, you can connect directly to the ma'am.
Yes. Or one more thing, I have an upcoming book for harnessing food system in microbiology, and you can also have access for this book and read every questions which you have now. Okay, in detail in that book, everything will be there.